Good morning. I'm Stefano De Monte from APE Research, a biotech company here in Trieste, and I would present some issues on biological and biomedical application for scanning probe microscope. Two things about the company. The company started in the previous century. Okay, since in 1999, APE Research is active in various sectors. The main sector is to design and to develop scanning probe microscope, a specific type of microscope that we will quickly give a look in a few minutes. Development of new investigation method in biology and physics based on this use of probe microscope is the main activities followed from our team that is based in Area Science Park, as you can see, and it's composed from many people, about 15 people from engineering experience. There are some biologists, some physics engineer, and so on. We, in general, develop scientific instrumentation for surface physics. Just to give you an idea what kind of resolution and type of instrumentation, I give you some example just to give you some idea about a resolution. Then we can see from the left some plant cells that have some resolution, some dimension that could be easily investigated by optical microscopy. Then if you go into the world of the nano, okay, we can approach some bacteria, some virus, proteins, about 10 nanometers resolution, and we arrive also to atoms from 1 angstrom to 0.1 angstrom. What kind of other instrumentation? You probably just know electronic microscopy is named SEM, scanning electronic microscopy. Okay, they have a typical resolution of 10 nanometers or something more. Our family of microscopes are the scanning probe microscope, and we have 3 different types. Then we start from an instrument called SNOME that is about 10 nanometer resolution, to FM and to STM that have atomic resolution. We are looking very, very, very near the atoms also. Then to give you an idea of the design of this kind of products, I present some short images of this type. From the left, there is a TRIA SNOME, then there is an IFM, and there is STM. What is the difference between these 3 kinds of instrumentation is the principle that they use to build, to have information and images. I don't want to go into the details, technical details, but I would give you some example. Another things on the right part of the slide, there is another new family that is called the scanning micro Raman. It's based on a different principle, and this means that our capability to integrate this other kind of technology within our instruments. Just one idea where we operate. We operate all around the world, not all, but the many countries. Obviously, we are based in Italy, we operate in Europe, but also in Northern Africa, also in the Far East, Korea, China, India, Saudi Arabia. And this is some country that we are able to follow, and we have some important customers that are really mainly academic customers, but really are something at the top of the art, the status of the art of university, as University of Strasbourg, PFL, just to give you some name, Queen's University in Belfast, obviously in Italy, and also in other countries that we have previously seen. These kind are user and, I said, partners, because some of them are partners in our research project. Our research project has a very important role in our activities, because if you can see some European projects that something are still running, we develop some strong cooperation with our partners. And since we don't know everything about every application of our instruments, we use this kind of project to increase our knowledge about specific topics that are the state of the art of some argument that also should be bio, should be material science, and so on. The other part, you can have a list of historical partnership starting from Trieste, obviously, CNR, former INFN task, obviously Area Science Park, obviously Elettra, obviously University of Trieste, and IIC Bangalore, because there is our common lab from Upper Research and their Institute of Material Science, and that is our external lab for India. Just to give you some information more about our main product that is called AFM. Okay, what is the characteristics? I can speak about this about 1 hour, okay? I don't want to be too much technical, but this kind of instrumentation is designed from our engineers and is able to have high sensibility. Then we can obtain absolutely high resolution. We are able to look at atomic step resolution. We can have also a good accuracy, a good sensitivity, and it is very flexible for the application we can have on it. Then this is also integrated with other parts that are all designed and built here in Trieste in our plants. Then this is a complete system on the left, and here there are some more technical working modes, contact, non-contact. Some specialists could understand, but in general you can see that we can have many ways to use this kind of instruments, and this means we can apply this instrument to many kinds of samples in different applications. This is a good flexible application. Then we can go to give an idea, a better idea about a possible application of this kind of instruments. Then there is also taken also from an article, atomic force microscopy should be also used for make some analysis on various classes of virus. Now virus, you know, it's very everybody's talking about virus in the last 2 years. We can obtain many information, topographic imaging of some part of this virus, to know better the morphology of some part of this virus, to understand better the DNA, RNA of this kind of virus. And in addition, we can have also some information about nanomechanical characteristics of the surface of this kind of virus. Then he is strong, he is flexible, the stiffness, and this is linked with the characteristic of the virus, of his life and his activities. Then this kind of instrument should be very useful also in this field. Another kind of instruments more useful for bio application is named scanning near field optical microscopy. SNOM is the acronym. It's less known, but it is based on some particular use of light. Then we have some integrated vision system. We have also an integrated inverted optical microscope that is more familiar for people involved in biotech. And we could also mix the other measurement of IFM with this. On the right side, you can see an example. Okay, that's this is a human sperm. Okay, then we can see some different characteristics using this special characteristic of the light in reflection and transmission or back reflection mode. Then you can have more information than traditional microscope. And also there are some publications made here also in Trieste. Other use of these two integrated measurement and analysis, both from IFM and SNOM, was done on asbestos fibers on cell. Asbestos is a big problem also in our region, you know well. And it's very important to understand how these asbestos interact with the human cells. Then you can see also in the image a cell, and also the lines okay, are the fibers of asbestos. And you can see and study how they should be inside the cell, how they enter and then make some disaster you well know. Different kind of analysis could give different kind of information that for the researcher should be very important. Also here there was a publication made here in Trieste specific. With our information, with our measurement, okay, scientists are able to have more information, we have just said, and are able also to make some good research, and this is a neuro image first page with our image of some specific okay sample, but also in this case you can see different kind of techniques on the same sample giving more information. It's very technical, okay, but from the last image C you can see that using the light with our instruments, you can have information from reflection, then from the first surface, and since you use the light, you could have also some information on the bottom part of the sample, inside, just a few nanometers under the surface, and we can discover what is under the surface. Just to have another example of application of activities of upper research, I previously said about some research projects, and the research project gives sometimes some good result. We have the opportunity to study bacterial biofilm. That is a big problem. They're present also in your washing machine, for example, and you know that if there is too much, also the quality of the washing was not good. They have also serious implication also on health and on the environment. Then we studied some new system, innovative system, that includes one sensor system to monitor this kind of biofilm. Just to give you an idea of the result, okay, we have developed a sensor system. In part is a quartz microsensor, and in part was an optical sensor. And this both system is able to give us the information about the growth of this biofilm and his type of bacteria present on it. That is a practical problem, a practical application, and also you can see also the development and also the final system, we have to notice that the application was also requested interest from Electrolux, and the result of this kind of research and application just existing should be also extended in different industrial sector that should be involved in food sector, biomedical, water, and so on. Then the applications are many. I thank you for your attention. This was just a quick example of our ability to manage instrument, to have more information from also the bio world, but not only for it. Thank you very much.